Okay. So good afternoon, everybody. Welcome to our immuno-webinars. Today, our speaker is Dr. Acácia Pinheiro Caruso Neves. Acácia is a friend, a neighbor here in our institute. And Dr. Acácia holds a degree in pharmacy and a PhD in biological chemistry. Both are from our university, Federal University of Rio de Janeiro. And Acácia has a period of postdoc at Johns Hopkins University in 2006. And she is currently an associate professor here in the Federal University of Rio de Janeiro at our institute, Biophysics Institute, Carlos Chagas Filho. And she's head of her laboratory. It's a laboratory called Immunology and Biochemistry of Parasitic Diseases, with a beautiful logo that I love the logo. Acácia is a CNPq fellow. She holds PQ1D fellowship. And Acácia has experience in biological science with emphasis on immunology, parasitology, and biochemistry. Malaria, that's the topic today. It's the main, I think one of the main subjects of her expertise. And so it's a pleasure to have you here, Acácia, with us. And thank you for accepting this on this kind of holiday that we have. So I thank everybody also that is here with us at this lunchtime on this Monday. So Acácia, please, the floor is yours.
Thank you for this invitation. Actually, I'd like to thank all the organizers for that. Let me share the presentation. Okay, can you see this?
Yes, perfect.
All right. So first of all, I'd like to thank all the organizers for this invitation. It's a pleasure to be here. So today I'll talk about the role of the renin-angiotensin system and also the kininogen system in the pathogenesis of severe malaria. So I'm not sure if you are all familiarized with the theme, so I prepared a little background about it. Malaria is a parasitic disease and it is caused by parasites from the genus Plasmodium. It is still considered a serious public health problem in the world, and it's also the leading cause of mortality. According to the last malaria report, there were 204 million cases in 2020. Also, among them, 627,000 deaths, and 80% of those deaths were in children with age below 5 years old. So besides all the efforts, the numbers are still alarming. And from this elevated number of cases, 1% turns into severe disease, which are responsible for other mortality and also morbidity associated to the disease, which imposes social and economic burden. So that's the importance of this disease in the world. So malaria is actually caused by different species of Plasmodium. There are 5 species that can infect the human host. So among them, Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, malaria, and NOLAS are the most common forms to infect the human host. The disease is spread all over the world, especially in tropical and subtropical countries, but the majority of the cases, I have problems here. Uh-oh, it's not going through. Hold on just 1 second. Okay. The majority of the cases and also the majority of deaths occurs in Africa country. Okay. So it's mostly because of Plasmodium falciparum infection. So Plasmodium falciparum life cycle is spread into different hosts, the mosquito host and also the vertebrate host. In humans, the disease in humans is split in, there are two stages, specific stages. The liver stage, which is asymptomatic, but it is responsible to generate merozoites that subsequently infect erythrocytes, initiating what we call the erythrocytic cycle. After several asexual reproduction, there is an elevation in the number of parasites. So parasitemia is elevated after several reproduction. And also the symptoms, malaria-specific symptoms occurs. So malaria symptoms, I don't know why it's not working. Excuse me. Sorry, people, I have problems here.
Don't worry. É a animação que tá com problema?
Não tem animação, eu só quero passar o próximo slide. E estava tudo funcionando, né? Que a gente testou.
Sim.
Deixa eu ver pelo Enter. Deixa eu tirar o pointer, se melhora, não sei. Here, vou por aqui. So, let's get back here. So there are several malaria-specific symptoms. All of those characterize the uncomplicated disease. So high fever, actually there's a cyclic fever that occurs, and also fatigue and ache, vomiting, chills, sweating. However, malaria caused by Plasmodium falciparum is specific and different. So the erythrocytes, when they host the mature forms of the parasite, they become different. So there are proteins from the parasite that are sent to the host membrane. So especially there's one, the PFMP1, that is expressed in the surface of host cells. And these cells now become sequestered in the endothelium that has receptor for these proteins. So in response, what we have, obstruction of the microvessel, the reduced blood flow, and also hypoxia. Actually, there are other several cell-cell interactions that occur in the blood tissue. So besides recognizing the endothelial cells causing sequestration and also microhemorrhage, there is also the recognition and interaction with other erythrocytes, infected or non-infected, causing resetting, and also interactions with the host immune cells, platelets, monocytes, neutrophils, T and B cells, are all of them activated, and they are responsible for a cytokine storm. So those are actually cell-cell interactions which are organ-specific. So it occurs in brain, lung, kidney, different organs. So they are specific immunopathologies. And because of this, falciparum malaria is associated to the severe disease. So here I call the attention to severe malaria, cerebral malaria, which is the most prevalent form of severe disease. And together with this, we can also, I would like to call your attention to acute renal failure, which is the second most common severe disease. Actually, it was not very well studied, but now it has a very large importance in the malaria disease. The thing is, in this presentation, we will talk about malaria pathogenesis. So I'll try to convince you that the renin-angiotensin system and also the kininogen system is also taking place in malaria pathogenesis. So we are going to start with the Plasmodium erythrocytic cycle, and then we move on to two severe disease, which is cerebral malaria and also the malaria-induced acute kidney injury. Okay? So the erythrocyte cycle is actually, it starts with a primary contact between merozoite and the surface of the erythrocyte, and then the merozoites reorientate to put together the apical junctional proteins with the host membrane, and then it moves into the cell and entry. The entry occurs and inside the erythrocyte, parasites is involved by a parasitophorus vacuole. And inside the erythrocytes, it differentiates and it develops into several daughter cells called merozoites, and it perpetuates the erythrocytic cycle. So blood parasitemia enhances, as also the malaria-specific clinical signs, as we talked before, occurs every time a schizont occurs the disruption of the erythrocytes in the schizont mature forms, okay? So a long time ago in the literature, it was very elegantly shown that several proteins from the host are moved, and they are appears in the parasitophil vacuoles. For example, the beta-2 adrenergic receptor, which is encountered in the membrane of the host cells, but is also in the membrane of parasitophil vacuole. Together with this, the G-alpha acid proteins occurs the same, but it's not true to the, for example, G-alpha Q protein, which is in the host, but it's not in the parasitophilus vacuole. So the thing is, when these people, they designed a peptide that target the G-alpha acid protein, and then when this peptide was in the culture medium, the entrance of the parasite invasion is inhibited. It occurs when they used the peptides, but it doesn't happen when they use the same peptide in a scramble sequence of the peptides. So it means that GPCR are important for the invasion, and also they show the maturation of the parasite. And our question was, if it's true for a GPCR, is it true for every GPCR? So what is the role of angiotensin II, which is a peptide that can bind to a GPCR in plasmodium erythrocytic cycle? So just to remember, angiotensin II belongs to the renin-angiotensin system, and it is formed by the cleavage of angiotensinogen by renin, forming angiotensin I, which is a substrate for the angiotensin converting enzyme, forming angiotensin II. Angiotensin II can either have has its action through the H1 or H2 receptor, but through the H1 receptor, there are several modifications that can occur. I call your attention to the pro-inflammatory response and also the pro-thrombotic response. Alternatively, angiotensin II can be other recognized by ACE2 and giving rise into angiotensin 1,7, which binds and signals through the MAS receptor, which is another GPCR. So to study the role of angiotensin II in malaria erythrocytic cycle, we used a reinvasion assay. So this assay, we started with a plasmodium falciparum culture. So falciparum is kept in cultures. So in culture flask, we can find all the forms of the parasites. So what we do in first step is the treatment with sorbitol to make the culture synchronized. And when at this stage we have ring forms, and after 24 hours, we have mature forms. In the mature forms, I mean, in the schizont stage, we add angiotensin II treatment. And after 24 hours, it's time enough to the parasites release and also reinvade. So what we call reinvasion is when we find young trophozoites inside the erythrocytes. We can see them easily by light microscopy. So in the first experimental group, we saw that angiotensin II, when added in the culture medium, it induced a reduction of ring formation in concentration dependent manner. So interestingly, we found H1, AT2 and also the MAS receptor in the membrane of the host cells of the erythrocytes. And angiotensin II effect of reducing the ring formation was reversed, not by losartan, which is a blocker of the A2-1 receptor. It was partially prevented by PGE-123-319, which is an antagonist of AT2 receptor of angiotensin II, but interestingly, it was totally blocked when we added A779, which is an antagonist of the MAS receptor. It suggested that angiotensin II could be degraded into angiotensin 1,7, and angiotensin 1,7 is the peptide that is reducing the erythrocytic cycle. So we compared the activity of angiotensin II with angiotensin 1,7, and both peptides reduced about 50%, 40% the ring formation. PKA activity was measured, and we found that both angiotensin II and angiotensin 1,7 were able to reduce PKA activity. And this activity, this inhibition, was reverted by A779. And to connect PKA activity to the erythrocytic cycle, we added imipaciclib to the cultures, which increased ring formation, but when we inhibited PKA, it reduced it. So our results together in this first part of the presentation, we show that angiotensin II somehow is cleaved in the culture condition, and angiotensin 1,7 through the MAS receptor reduced PKA activity that inhibited erythrocyte infection. This was very interesting. We were fascinated with our findings, but our question after that was, what is the role of RAS in the outcome of the disease? So those were in vitro studies. And then we go through and started to study cerebral malaria. So let's talk a little bit about cerebral malaria. So cerebral malaria occurs in 1 to 2% cases of infections, but it has high rate of fatality. 15 up to 20% of the cases succumbs to death. And when patients are recovered from the disease, they can still have long-term neurological sequelae. So the disease is defined by an unbrowsable coma in the presence of PF parasitemia, Plasmodium falciparum parasitemia, with no other known cause of neuropathology. The thing is, the cerebral malaria pathogenesis is not well defined, is not completely understood. So it seems to start with the sequestration of the parasite, which induces endothelial activation. And endothelial activations increases the expression of molecules which are involved in adhesion. I mean, ICANN1 and MHC class 1, for example. And then parasites are much more sequestered. Another thing that can happen is the increase in cytokines and chemokines expression, which put together here in this milieu cells from the immune systems. In a second wave of chemokine production, CD8 positive T cells that were primed in the spleens comes into this milieu, and they are activated by in an interferon manner because of cross-presentation of parasite antigen from the endothelial cells. So CD8 T cells, they have here locally effector production of perforin and granzyme, which are responsible to the disruption of the BBB barrier. I mean, apoptosis of the endothelial cells. So how proteolytic circuits could match to this mechanism? So I call your attention to activation and the control of the biology of CD8 positive T cells. Well, to study this, we use the experimental cerebral malaria model using C57 black 6 mice, which are injected with Plasmodium berghei anca strain. So this parasite induces in mice a disease that resembles the human disease. It's not exactly the same, but clinically similar to what happens in human hosts, okay? The thing is, when black 6 mice are infected with this strain of parasites, they have parasitemia around maximum 20%, something like this, which is not that high when bulb C mice are infected with the same strain. Parasites, in black 6 mice succumb to death in a window that can vary from day 7 to 12 days after infection, which doesn't happen with bulb C mice. And also, P. berghei anca induces in black 6 mice edema, cerebral edema, which can be measured, easily measured by Evans blue assay. So using this model, I'm going to show you some results. So the renin-angiotensin system was in the last years, more than 10 years ago, they were shown to be present in different cells from the immune systems. Actually, T cells, they have endogenously production of angiotensin II because they have all the components of the renin-angiotensin system. So our question was, what was the ability of angiotensin II? Is there a role for it to control or somehow influence the outcome of disease, of cerebral disease? So we used the experimental malaria model, and mice were infected or not, and infected mice were divided into other 3 groups: vehicle treated or losartan and captopril treated. So losartan is the blocker of the H1 receptor, an antagonist of H1 receptor, which means that it blocks angiotensin II signals. And captopril is an ACE inhibitor, so it inhibits angiotensin II formation, okay? So we used these 2 pharmacological approaches to detect the influence of angiotensin II and even the H1 receptor axis signaling in this disease. So what we found was that losartan and captopril reduced activation of both CD4 and CD8 cells measured by CD69 expression. Also, losartan and captopril reduced interferon production and IL-17 production by CD4s, and also reduced perforin production by the CD8s. So it seems that angiotensin II signal through the H1 receptor is important for induces the effector function and activation of T cells. Interestingly, when we try to detect the presence of these cells in the brain of the mice, we saw that captopril were able to reduce the homing of the cells to the brain, which was not observed by losartan. However, besides we did not detect here an effect for losartan, cells that went to the brain, they were less activated. More, the activation was more, inhibited by losartan than captopril values. So this phenomenon did not imply very avidly in parasitemia or survival of the animals. We can see a difference. Actually, parasitemia is very little reduced, but it still has parasitemia. So the survival curve was not that improved. However, it's very important to mention that angiotensin II signal here interferes in T cell activation. And what is the role of this signaling in antigen-specific T cells? So we move forward and go through this mechanism in collaborative work with Dr. Fidel Zavalas from the Johns Hopkins University. So there they have transgenic mice which has possessed, these mice, they possess CD8 positive T cells, which are antigen specific. So these cells from that mice, they recognize a peptide, the synthical peptide, which is present in the CS protein from P. bergei ANCA sporozoites. So those mice, they were crossed with H1 receptor KO mice. So then we generated mice which were malaria specific and also deficient of the H1 receptor. And then to observe and follow T-cell activation in the cells, we used the immunization of these mice with the CS, the sporozoites, sorry. The sporozoites were obtained from the salivary glands of the mosquitoes. They were attenuated by irradiation and used to immunize the mice. So what we did was we got CD8 positive T cells from white type or from knockout mice, transgenic knockout mice. Those cells were adaptive transferred to recipient naive mice. And they were immunized with the sporozoites, and then we followed the CD8 positive T cell response from day 3 up to 32 days after infection, just to follow the whole wave in the response. So first, we detected that the clonal expansion of the cells were attenuated when we have knockout CD8 T cells. So we see from day 3, no different, but since day 7 to the end, the clonal expansion was reduced. Together with this, we detected that IL-2 receptor expression measured by CD25 and CD122 was reduced in H1 receptor knockout, and also the production of IL-2 was also reduced in the absence of H1 receptor. So those results suggested that angiotensin II is produced and binds to H1 receptor, and it is important to regulate clonal expansion, IL-2 expression, and also the IL-2 receptor expression by CD8 positive T cells. So together we saw a little difference in parasitemia, which is very similar but in comparison to what we detected before with the pharmacological treatment, and also survival. So together, these results suggest that CD8 positive T cells, when they are activated by malaria peptides, they can produce angiotensin II. Angiotensin II can bind and signals through the H1 receptor, and this signal is important for activation, CD69, CD25 expression, proliferation, differentiation of the cells to acquire effective function, and the production of cytokines, especially IL-2, TNF, perforin, interferon gamma. Obviously, I did not show all the results that helped us to build this model. Okay. All right. So when we used the inhibitor of ACE, our objective, our goal was to reduce angiotensin II formation. However, ACE inhibitors, actually ACE are a connection between two systems, the renin-angiotensin system and also the kininogen system. So here in KKS brain, what we see is that high molecular weight kininogen can be cleaved, giving rise to bradykinin. So plasma kallikrein is the enzyme that cleaves the molecular high weight kininogen, giving rise into bradykinin. If ACE is present, bradykinin can be degraded into inactive fragments. However, when we use ACE inhibitors, in one hand, we have, we abolish angiotensin II formation, but in one hand, we increase bradykinin levels in the milieu. And bradykinin is a peptide which induces pro-inflammatory mechanisms, very well known for this. So in the next step, we were interested in the effects of bradykinin in our systems. So first of all, in the cultures of Plasmodium falciparum, in the supernatant of the cultures, we could detect bradykinin levels in this culture. And also, we detected high molecular weight kininogen very close to infected red blood cells, but not together with non-infected cells. Together with our observations, there is also a relate in the literature. The group of Bagnaresi et al., they also recognized that high molecular weight kininogen was detected by parasites. Actually, it is collected from the milieu. Parasites, proteases can cleave the high molecular kininogens, giving rise to different peptides, among them bradykinin. So our next question was, what is the effect of bradykinin in this complex cerebral malaria pathology? So we decided to study if it induces pro-inflammatory response. So we decided to study brain endothelial activation, maybe the increase of parasite adhesion to the endothelial cells and its consequence. So for this, we used co-culture assay. So first we have the Plasmodium falciparum culture kept in vitro. We collected the supernatant of this culture after Schizon disruption, what we call conditioned median. So the adhesion assay of infected erythrocytes to the endothelial cells was observed by treating the human brain microvascular endothelial cells with culture supernatant overnight treatment. And after that, we put together infected red blood cells. And then we count how many infected cells are in contact with HB max, which is our adhesion assay, okay? So we detected that the condition median increased adhesion of infected erythrocytes to HB mac, which actually occurs without any treatment, but it increases the basal adhesion of these infected erythrocytes, okay? The effect of the conditioned median was very comparable to the effect of bradykinin when it is added instead of the conditioned median to the co-culture assay. And also the effect of both culture median or bradykinin was reverted or prevented by HOE and DLBK. HOE is the blocker of the B2 receptor, and DLBK is the blocker of the B1 receptor of bradykinin, showing that the signaling of bradykinin induces adhesion of infected erythrocytes to the endothelial cells. In the next step, we trying to observe how was the effect of bradykinin in permeability of the endothelial cells. For this, we used it in Transwell system. The treatments were added in the upper, let's say, upper chamber. BMAC monolayer is cultured here. And then after overnight treatment, we add BSA fluorescent actually as a tracer. So what we would like to do is detect the fluorescence in the upper chamber, okay? Upper bottom chamber, right. So albumin permeability was detected when cells were treated with both BK and also the conditioned median. But this phenomenon was blocked when we used the receptor antagonists, showing that this signal was important to induce the disruption of these monolayers. So we go further and performed some immunostaining assay. Here we stain ZO1 and beta-catenin, showing that these proteins are in control situation in the surface of the cells, especially among two cells, because they are part of the interactions, the junctional endothelial proteins. But when we treated the cultures with the conditioned median, there is disarrangement of these proteins and cells become detached. And this phenomenon was partially blocked when we used HOE to block the B2 receptor. So they kind of try to assemble again. And together, those results shows that maybe we can find bradykinin in very low levels in the culture medium in normal condition. But when during infection, somehow the bradykinin levels is increased, and bradykinin induces first the adhesion of infected erythrocytes to the endothelial cells, and also the disruption of the cell junctional ranges, inducing edema and BBB disruption. Okay, if the pathogenesis of cerebral malaria is not well understood, you will cannot imagine the studies concerning malaria acute kidney injury. It's very rare in the literature. And what we know about malaria-induced acute kidney injury, it is present in at least 50% of several malaria cases. It is also associated to poor prognosis and death, especially when it occurs together with cerebral malaria. I mean, as a component of a multi-organ dysfunction, but it can also occur as a unique pathology. So the disease has two components: a glomerular injury induced by deposition of immune complexes of mononuclear cells infiltrations, and a tubular interstitial injury, actually induced by infiltration of mononuclear cells. However, the role of T cells in this scenario is not well understood. Actually, is not studied. Actually, very rare in the literature. Okay. So we decided to study the role of CD8 positive T cells in MACI pathology. So we used three different approaches. The first one is the treatment with FTY-720, which is a drug that inhibits the egress of lymphocytes from the lymphoid organs. We used also adaptive transfer experiments and also depletion of CD8 positive T cells. So using the FTY treatment, so treatment starts right after infection. So mice were infected with Plasmodium berghei anca, and then the next day the treatment starts, and it occurs until day 4 post-infection, and day 5 we collect organs and also urine and blood samples for analysis. Okay. So this is to show you that our FTY treatment worked. So here we have no influence in parasitemia levels, but it reduced drastically CD4 T cells and also CD8 in the blood circulation. Okay. All right. So when we detected the functional parameters of the kidney, so what we detected, proteinuria and also the ratio of protein and creatinine, which is increased during infection, they were reverted when mice were protected from proteinuria when they were treated with FTY. Also, a marker of tubular damage, gamma GT, which is detected in the urine, and it's increased by infection, it was reduced by treatment. When we detected parameters of the glomerular function of the kidney, we didn't see any difference, which means that glomerular injury that occurs in malaria, it was not abrogated by FTY treatment, which means that lymphocyte may not has a role in this arm of the disease, kidney disease. So after this, we decided to do adoptive transfer experiments to detect the participation of T cells. So we have naive or infected mice. From these mice, they were donor of splenocytes. From this spleen, we collect T cells, and T cells were CFST labeled and transferred to naive mice, okay? After adoptive transfer, at day 3, we detected CFSC labeled cells in the kidney, in the spleen, and also in the brain of infected mice. Okay, this is to show you that when we transferred the cells, we did not transfer parasites. The cells were, clean and avoided of any parasites. Okay. So proteinuria and UPCR, as we saw before, was reduced by adoptive transferring T cells. Gamma GT, urinary gamma GT, was recovered, but there was no difference by transference, no difference in glomerular markers. It occurs in functional parameters, but also in the arrangement, the structure of the kidney. So we got, hold on, let me move the screen here because I need an information which is, okay, got it. So when we detect the structural assembly of the kidney, so interstitial space, which is increased in infection. Also, interstitial cellularity is increased by infection. And when we see, when we did adoptive transfer, when we see a recovery from the tubular structure, but not a recovery of the glomerular modifications. I mean, there is space, a huge space in Bowman's capsular, and also glomerular cellularity. Move on, please. Trying to move the slide. Hold on.
Stopped again.
Oh, okay. Here. All right. So besides, we detected several pro-inflammatory cytokines, and IL-17, IL-6, and interferon gamma was increased in the kidney of the mice that received CD8 positive T cells that came from infected mice, but not in mice that were adoptively transferred with naive cells. Okay, TNF, alpha, and IL-4, we didn't see any difference. Moreover, CD8 positive T cells were increased in the renal tissue, and also there was an increase in perforin production when we that were detected in mice that received CD8 positive T cells, actually T cells that came from infected mice. Okay. The depletion of CD8 positive T cells did not interfere in parasitemia. Actually, there is a slight difference at day 5. Okay, but what we see here is that CD8 positive T cells were, this is, okay, I'm sorry, we changed it to the depletion. I'm sorry. So I have a lot of things in my screen. I cannot follow this slide because of those things here. Okay, I'm back. So after this, we perform a depletion of CD8 positive, CD8 cells specifically using antibody. Okay, so after infection, mice were treated with anti-CD8 on day 2 and day 3 after infection. And then on day 5, we did the analysis. What we saw here is the depletion worked. So we saw a reduction in the number of CD8 positive T cells in the kidney and also in the spleen of mice, infected mice. It slightly reduced parasitemia, but it was on day 5. Okay. Okay. So the depletion of CD8 positive T cells actually reduced proteinuria observed during infection and also reduced the ratio protein to creatinine in the urine. Reduced also the marker of tubular damage, gamma GT, but it did not change the markers of glomerular disease, glomerular injury. So together, we can conclude with these results that in the literature, we can find that rupture of infected cells actually parasites components and parasite itself induces glomerular injury because of an inflammatory response that occurs in the glomerular region. So here, based on these results, we add another piece in this puzzle, adding that an increase of pro-inflammatory cytokines with CD8 T cells activation induces tubular injury, which induces proteinuria in infected mice. So I'd like to acknowledge all the people in the lab that worked together in these results. And specifically, I'd like to acknowledge Dr. Salsa Caruso Neves, Dr. Julio Schaffenstein, and also the collaborators from Fiocruz and from Case Western Reserve University. And obviously, I'd like to also thank the agencies that make possible this all this work. Thank you all.
Thank you, Acácia. Thank you very much. Let's talk.
Let's talk. Yes.
Let's talk.
I need to stop, right? Hold on.
Yeah, I think you could. I'm trying to. Oh, no, it's done.
Should I? May I stop? Sharing?
Yeah, you can.
Okay.
I think you can. Yes.
Okay.
So now
I'm sorry, I was a little bit confused because there are several pop-ups in my screen. I couldn't see the slide very well.
I had the same problem when I did a presentation on UFRJ, so I completely understand. And Zoom is new for me, so
yes, it is.
So that's okay. Thank you very much. It was a beautiful presentation. And Acácia, what we usually do first, it's open for questions by students. Okay. So to incentivize that, I know that Miriam has her hand up there. So I'm just going to check if we have any question from students. Miriam, can you help with that? Do we have questions from
Not
students?
Yet.
Not yet. Okay. So let's start with Miriam. Yeah, let's start with Miriam.
All right.
So then the students get relaxed and start doing questions. Okay, Miriam.
First of all, congratulations on all the research. I wasn't very familiar with your research, so I'm really excited and very, very interesting, the data you showed.
Thank you.
I have a related question. I saw that the proposal is that when looking at the first part of your talk, that through AT1 receptor, you'd be emphasizing the CD8, the T cells would emphasize the expression of CD25, CD69, all the activation markers and IL-2. And I'm not super familiar with the type of signaling, but I saw that calcium might be upregulated through the GPR receptor. And I wonder if if you plan on looking on NFAT or a calcium influx, and if you think that could be a co-stimulatory, could be playing a role of co-stimulatory molecule to help on the T cell response.
All right. Yes, totally possible. Actually, we didn't see that. We didn't study calcium levels inside the cells. group in São Paulo that actually studied this. And they saw an increase in calcium signaling. And actually it is involved somehow with I believe, I don't remember now the protein, but it's a protein from the junctional system, and it influences the BBB disruption. So for parasite itself or inside the red cycle, the red blood cells, I'm not sure if there is any report in the literature, but yes, in that milieu concerning the BBB disruption, yes, it is important.
Thank you. Yeah, I was wondering on the T cell if you'd test using cyclosporin A, or if you could interfere with drugs on that end and also get some response.
Very nice. Yeah, we always want to, but we never can do everything that we think about it. Yes, it's a possible situation. And we are now looking into different, it's not in the A2-1 receptor signaling, but we are curious about the bradykinin signaling as well. So maybe it's something that we can think about it in our next steps. So we are working in how bradykinin can induce BBB disruption or adhesion of infected cells to the endothelial cells. So it's a work that we are doing together with Dr. Julio Schaffenstein, and also together with a group from the Cleveland University, the Case Western Reserve University in Cleveland. So yeah, it's promising. So I didn't bring these results. It's something for, next time.
Thank you. Chris, check the YouTube as well, but there are no questions yet.
No, we have a question from the chat, the room chat here in Zoom. It's from Teresa Ramalho. I don't know if Teresa wants to open the microphone, or I can just read the questions to Ana. So Teresa is asking, she's saying nice talk, and asking, do you know the mechanism involved, the mechanism that induced the migration of CD8 cells from infected mice in the brain, spleen, kidney of naive mice in this adoptive transfer system?
Okay, no, we don't know now. I cannot tell you, but I have a clue. So actually based on what happens in the brain. In the brain, endothelial cells, actually they do a cross presentation. So this is what implies to a consistent activation of CD8 cells in that milieu. In renal tissue, it was never shown before. However, there is a paper in the literature that detected T cells in the interstitial space near the tubular cells, but they could not find CD4 T cells. They did not say anything about CD8s. But there's another paper, another manuscript, actually studying a different disease. It's a kind of lupus systems. Actually, they have glomerular disease, very similar to what is found in malaria, and also tubular damage. So in this model, they could detect T cells close to the tubular cells. And actually they detected endothelial cells presenting antigen to the CD8 T cells. And actually it resembles the mechanism that occurs in the brains of infected mice. We don't know if it happens. And actually our system, the system that we use it, we cannot imply this with that because we did not infected the mice after adoptive transfer. So this is something that we are planning to study in the near future. Thank you for the question. Did you have the chance to check the activation of disease during the treatment with losartan? No, I didn't have that. Yes, this is nice.
Well,
it's a possible explanation, actually. So using the transgenic system, we saw the effect of H1 receptor in the cells. But if there are other results in adoptive transfer that we can imply that disease could be also interfering when we transfer. So we did the transference of transgenic cells to naive mice, but we did the other way, naive cells to transgenic mice. So we could detect that maybe disease could be affected, but no, we did not study that. And you, Chris?
Thank you. I want to ask. I have
Lucas also has a question, Chris. Lucas Bolini.
Oh, Lucas, let's hear from Lucas. Lucas Bolini.
Hi, Lucas.
Hi, everyone. So I think my question is also kind of related to this
Lucas.
Question.
Sorry, Lucas. It's a little bit too. The volume is too low.
Yeah, the volume is very low.
Let me try.
In this position, we can talk. Yes, nice.
Is it okay?
Yes.
So my question is kind of related to Teresa's question. It's also about recruitment of T cells, but looking at it in another angle. So it's kind of well known that endothelial activation also promotes CXCL10, and I think also CXCL9 secretion by the endothelial cells, which also helps to recruit T cells. And I was wondering, since you have shown us that both the renin-angiotensin system and the kinin system interfere somehow either with the endothelial activation or with the T cell recruitment, if you have checked if either one of those systems could also interfere with endothelial production of CXCL9 and CXCL10.
Yeah, good question. So cytokines are also important for that, yes. And so I didn't bring all the results, but yes, we using the experimental cerebral malaria model, we could detect a reduced production of chemokines. And but in the renal disease, we didn't have the chance for that. So yes, it could be another explanation for the recruitment of the cells to the specific organs. So we detected that for the brain, but for the renal tissue, we don't know.
And in the kidney, did you see, other than T cell activation, did you see if there was a difference in the phenotype or in the recruitment of other leukocytes, like activation of macrophages, recruitment of neutrophils, or something like that?
Okay, no, we didn't, but actually macrophage and neutrophil activation are in the literature, okay? So it's not in the literature if they have actually the renin-angiotensin system involved in this process. Actually, we are now moving to study the renin-angiotensin system participation in this context of T cell homing and activation and their role in the renal disease, okay? And maybe it's in the next step, but we have a previous work showing that losartan treatment of mice in using the experimental cerebral malaria reduces the renal damage. So why? We didn't know. Maybe it's because of T cell activation and etc., but we didn't check yet. Okay.
Thank you.
Hi. So, first of all, I had love to tell you how I liked the immunopharmacologic approach that you got. So, captropil, losartan, Roy, I love it. So that's a feature that it's very interesting in a parasitological study like that. But because of that, and because you moved from different models and different antagonists and inhibitors, I got a little bit lost concerning what's the receptor that you are really, betting on. Because in the first moment, it was mass, the name of the receptor is mass. Then I saw 82, then 81, then DK1. So what your impression for all this? What you have like,
I could tell you there are more. I could tell you there are more receptors to tell about. Yes, so those systems are very intricate. So it's kind of difficult to, how can I say, pick one and decide this is the one that is the responsible for this or that, event. So the thing is, the pathogenesis of severe malaria is very complex. So we have different, we can actually attack in different points of the response. So in the first part of the presentation, I was trying to attack the erythrocytic cycle of the disease, which is where everything starts, correct? So in this situation, I can tell you that MAS receptor, which is the receptor for angiotensin 1,7, can reduce parasite infection. All right. But actually, we have more results. I can tell you that MAS receptor is not alone. It is together, and it kind of interact with the bradykinin receptor in the membrane of host erythrocyte. So, and it doesn't matter the molecule that is binding, bradykinin or actually another peptide or angiotensin 1,7, the response is reducing the erythrocytic cycle. It makes sense because there is another...
Just to check, Ana, this is human cells, isn't it? When you are talking about the
human. Yes. The erythrocytic cycle, when we studied that as human, was modern falciparum. Yes. The thing is, there was a paper also showing that there is a population in India. They have a kind of mutation that results in high levels of angiotensin 2 levels in the circulation. And those people, they are kind of protected from severe disease. They just have mild disease, uncomplicated malaria. So based on that, it makes sense. But however, it's not angiotensin 2 signaling through the AT1 receptor. It's probably angiotensin 2 signaling through the AT2 or angiotensin 2 that is degraded, and angiotensin 1,7 is signaling through the MAS receptor. So it's complicated because all those peptides are kind of converting into a new one. And it's hard. It's hard to say. In inflammatory response, I can tell you that is the H1 receptor because of the transgenic mice that gave me that very clear evidence that H1 receptor is involved in that. Hi, Bruno.
Let me ask you two small questions. Just to confirm, the conditional medium assay, the transfer of the conditional medium, these were human too?
Yes.
This is human too. It's very interesting. So do you have, I know that you found bradykinin on that, but do you have, a better characterization of this?
The source of bradykinin, yes?
No, not the source of the bradykinin. The other players that are together with bradykinin in this medium, the soup.
Well, we detected by mass, mass spectroscopy, we detected angiotensin 2, angiotensin 1,7, bradykinin, and appear other peptides that I cannot tell you what they are exactly. But we found those peptides that I talked about.
Okay, great. Thank you. Thank you.
Okay.
Let Bruno ask questions. Thank you.
Hello, Acácia.
Hi,
nice to see you. I must confess that I got just in time for your last slide, so I'm going to ask a question anyway. because
Okay.
I'm curious about, but maybe something that you already shown in the animal models. I would guess that you were showing that one of your last slides, it was a model showing the kidney injury, and you probably did the one infection and its brain, its course on this animal. But in the human population, we usually have reinfection of the population. How it would play out with the kidney injury? There is any data that these repeated injuries are causing more damage to the kidneys, or what's going on there?
What I can tell about it is that, so cerebral malaria is more common in children. The kidney disease appear to be more common in adult people. However, in the last years, renal disease is actually a second trigger to death when it occurs together with cerebral malaria in children. And actually, when these two pathologies are together, it's very hard to recover the children from the disease. So when they do not die, actually they stay with long-lasting disabilities, neurological disabilities. So there is a participation somehow, a crosstalk between renal disease and also cerebral malaria. I cannot tell you what exactly is by now, but you asked me about repetitive malaria, right? Well, actually, 50% of the patients can appear with renal disease. However, it is treatable somehow. Patients can recover, but what happens is that when these patients, when malaria is repeated in these patients, they can go to severe kidney disease. So the chronic kidney disease is achieved more avidly than, in the first malaria infection. Okay, so it turns the patient more, how can I say, sensitive to go through the severe kidney disease.
Right.
And, end stage kidney disease.
And it's something that's, I've been following the news here about all the floods in Pakistan. And they had these huge rains like a couple of months ago, and they are still dealing with lots of areas that are flooded. And I saw on the news yesterday showing all these children that were dying of malaria. So it's sometimes people think, malaria infectious diseases is something that is from old times and something that is coming down. But with the climate emergency that we have now, and it will probably get worse. We'll get a lot more malaria, and there is still a need to really understand this disease, to understand all its impacts in its all different organs. And that's another thing that I really want to congratulate you to bring to our attention the kidneys, because we usually think about in malaria models, we only think about the brain, sometimes the lungs, not that much beyond that. And it's very nice that you brought to our attention all this damage also to the kidneys, and how important the kidneys are. Celso should probably be agreeing with me now that kidneys are really important organs with all these chronic diseases and systemic inflammatory responses. And I thank you very much for bringing these to our attention and all the work you have been doing in this area. Thank you.
Thank you for the comment. Yes, the renal disease associated to malaria was really neglected. And it's very important to keep an eye on this too. Thank you.
Anna, Chris is having a little bit of a technical issue. Bruno, yes. And so I'm gonna call Luis Juliano, who has a question. Would you like to open your audio?
Yes, I'm here.
Yeah, I can see you. Thank you.
Bom, como eu sou o mais velho em todo este conjunto, eu vou me dar o direito de falar em português.
Claro, professor.
E eu estou aqui, fiquei sabendo desse seminário, primeiro, há uma semana, pela Ana Paula de Pique, que por acaso é minha nora. E aí, quando eu vi o seu nome, Ana, não preciso te dizer que eu fiquei a semana toda discutindo com o meu amigo Júlio Schafstein sobre o seu trabalho.
Muito bom.
Colocou aqui, porque nós discutíamos, e você conhece muito bem, o senhor Júlio Schafstein, né?
Conheço bem.
Desde 1995, 92, quando ele chegou no meu laboratório, aqui na Escola Paulista de Medicina, um tubinho dizendo, olha, dizem que essa proteína aqui é uma enzima proteolítica. Eu do trabalho, peguei uma amostrinha e, na hora, é uma protease. Era a tal da cruzipaína do Júlio. E aí começou um trabalho, né, das cascatas proteolíticas, certo? Nas doenças parasitárias.
Qual eu sou super fã.
Você conhece muito o Júlio e toda a linha lá da UFRJ, nesse tipo de trabalho. E primeiro, parabéns, você ao vivo tem uma apresentação excelente, didática, e muito claro tudo que você colocou, e bastante entusiasmante.
Obrigada.
As cascatas proteolíticas, certo? São fundamentais, né, nas doenças infecciosas, né? Quer dizer, você infecta uma célula com qualquer um desses parasitas, Leishmania, Cruzi, Plasmodium, seja qual for, ele vai entrar, né, na célula que for e vai usar a ferramentaria proteolítica dele, que ele precisa, né, seja para se alimentar, né? E o Plasmodium tem ferramentas proteolíticas, né, impressionantes. As falcipaínas 1, 2 e 3, há um negócio extraordinário, né? Eu fui introduzido à malária aí na USP, né, pela Célia Garcia, né? Até esperava você participar hoje desse seminário. Célia eu conheci também no fim dos anos 90, né? E foi quando eu fui introduzido à questão da malária, né? Como a gente já estava vendo com o Júlio a tripanossoma, depois as leishmanias, né? E nesse grupo todo, né, só para notificar aqui, né, o Júlio e eu participamos de um projeto europeu, né, financiado pela Comunidade Europeia, em leishmanias. Por que a Europa interessada em leishmania? Porque a mudança da temperatura, né, global, isso no começo dos anos 2000, estava fazendo com que os mosquitos começassem a invadir o sul da Europa.
Exato.
E essa cascata proteolítica, essa história toda interessou a muita gente, né, na Europa, e nós, Júlio e eu, participamos desse projeto. E foi interessante, né? Não tenho tempo aqui para mostrar, mas tem uma foto do ano 2000, em Bordeaux, em que o time, né, dos interessados em proteólise e em parasitas estavam lá. E os nomes que hoje, né, estão na indústria e tudo mais. E qual não foi a minha surpresa que o Morten Medal estava nessa, né? Ganhou o prêmio Nobel esse ano.
É isso.
Quer dizer, então, o tema, tá certo? Aliás, quem foi... o Morten era um moleque ainda novinho, né? Eu conheci na Espanha, em 1990, e justamente ele se interessava por um jeito rápido de mapear especificidades de enzimas proteolíticas. Né? Eu que introduzi ele nas questões do Júlio. Né? Você imagina, você conhece muito bem a confusão que deu. Né? Ainda bem que o Julinho não está aqui, eu posso falar mal dele.
Pode falar.
Foi muito produtivo, entrou o Alves, né? Schmeier, né?
Alves Schmeier,
Então, foi sempre muito útil, muito estimulante, né? Esse tipo de operação de trabalho, né? E foi com muito carinho e muita atenção, né? Que eu fui lá, fiquei esse tempo todo olhando, baixei os seus trabalhos. Como eu não podia falar com você, eu fiquei...
Poxa, obrigada, professor. Fico lisonjeado, sabe?
Você briga com ele.
Poxa, não, eu agradeço muito. Aliás, o Júlio, ele realmente me dá muito apoio, sabe? Ele está sempre junto comigo, eu discuto muito com ele os meus resultados, porque é um assunto comum.
Sim.
E ele sempre me dá muito apoio e me bota para frente para fazer mais e mais. E foi através dele, ele me convidou. Na verdade, foi um kainin meeting que ele me chamou para ir. Eu fiz uma apresentação e nesse kainin meeting o Dr. Alvin Schmeier era o host. E aí foi quando eu consegui chamar a atenção do Dr. Alvin através dos meus resultados. E hoje a gente está fazendo junto. Se Deus quiser, a gente está conseguindo alguns resultados bastante promissores, que tem um pós-doc do laboratório que está lá no laboratório dele, tentando avançar nessa parte, né? Como que esses peptídeos são gerados. Aí, pegando um pouco da atividade de falcipaínas, a gente conseguiu uma bergeipaína, que é um plasmodium bergei também modificado. Então, a gente está tentando avançar se a origem, né? Tentar mostrar se a origem de bradicinina no nosso sistema depende dessas proteases, né, do parasita. Não sei se isso vem do plasma que a gente suplementa a cultura ou se é no próprio ensaio, né, de co-incubação com a célula endotelial, se de alguma maneira tem uma exposição ali do cininogênio de alto peso molecular e o parasita se aproveita, né, disso para conseguir mecanismos de aumentar a adesão, de aumentar a inflamação e progredir com a doença. Poxa, obrigada pelo comentário, professor, gostei.
Eu já sei, inclusive vai ter um momento que nós vamos conversar, você, eu e o Júlio. Né?
Tá joia.
Já está planejado esse tipo de coisa, porque eu tenho que olhar a cascata de...
Tem, né? Isso, eu vou muito mais para a questão do fenômeno, né? Mas é claro, isso daí já entendo que é uma pergunta que a gente precisa responder.
Para ir na 2 e 3, gera bradicinina.
Então, in vitro a gente conseguiu, colocando falcipaína em contato com o cininogênio, a gente viu que gera bradicinina.
Então, isso aí vem desde...
In vitro, com a proteína purificada. No sistema ainda com parasita, a gente ainda não conseguiu fazer.
O meu trabalho com o Júlio foi sempre assim, eu faço in vitro e passo o resultado para ele. E eu falei, não quero ouvir o que você vai fazer da biologia. Eu sou um químico. Embora eu sou formado na Escola Paulista de Medicina, sou um médico de diploma, aposentado na expulsória. Continuo trabalhando. Tá certo?
É isso aí.
É química. Eu gosto de lidar com os químicos.
Entendi.
A parte biológica é problema seu. Agora eu estou dizendo, o problema biológico é de vocês. certo?
Certo.
O sistema hipertensor renal foi onde eu fiz meu doutorado, né? Quer dizer, então, eu só queria fazer agora alguns comentários que chamam a atenção. Quando você usa o sistema hipertensor, estuda pressão arterial, sistema hipertensor em rato, o sistema renina-angiotensina em rato é diferente do humano. Na evolução, teve uma bifurcação, né? Quando chegou nos humanos, o sistema renina-angiotensina divergiu do humano. Então, é um problema. Você transladar dados de roedores para o humano, tem hora que você bate a cabeça na pedra. Entende? Então,
Eu entendo. Existem...
Tomar cuidado com isso, né?
Sim.
Então, estou chamando a atenção para especialmente para esse ponto. Segunda coisa que eu acho interessante e sugiro olhar, né? Eu comecei a olhar isso depois que eu fiz o seu trabalho. Uma coisa que está entrando é o levantamento epidemiológico da hipertensão na África relacionado à malária.
É um mapa que se sobrepõe, né, à infecção por malária. Onde você tem hipertensão, você tem malária prevalente. É impressionante.
Então, aí você tem um trabalho complicado, porque as casuísticas, o relato dos dados ainda são muito pobres lá. Mas já existe duas revisões, uma eu consegui, a outra eu pedi para o autor. É coisa desse ano, desses meses.
Olha, que legal.
Uma revisão sobre a questão da hipertensão e malária. Né?
Sim.
Então, isso é uma coisa que tem que olhar, porque isso pode te
Sim.
ajudar e ter muita coisa interessante aí.
Isso aí.
Né? E a terceira sugestão, né, quando você está olhando, e aí eu concordo, a sua contribuição é incluir o rim nessa história. Eu até na discussão com o Júlio, eu falei, a melhor coisa que eu gostei do trabalho foi a parte da fisiologia renal que ela colocou. Por quê? E aí vem a sugestão, quer dizer, você se aproximar do pessoal que faz transplante renal. Por quê? Porque o transplante, você tem as glomerulites, né? A resposta imune o grande problema do transplante renal, como qualquer outro órgão, é a questão da rejeição, né?
Sim.
Então, quem faz no transplante renal se acompanha a evolução da resposta renal, né, por biópsias. Então, aqui na Escola Paulista de Medicina, nós temos o Hospital do Rim, que faz nada menos, nada mais que 1.000 transplantes por ano. É o hospital que mais transplanta no mundo. Então, o que você mostrou, né, essa resposta inflamatória do rim e tal, quer dizer, o pessoal que faz transplante pode te ajudar muito. Inclusive, o patologista que lê o glomérulo. Porque você sabe, o glomérulo você tem um podócito, você tem... Enfim, o glomérulo é complicadíssimo.
Exato.
E a resposta inflamatória do glomérulo dados são as mais diferentes, certo? Então, acho que esse é um ponto rico do seu trabalho e que você ganharia bastante procurar pessoal. Aí no Rio tem pessoal que faz transplante e mostrar, inclusive, essa foto que você mostrou, né, do histológico do rim, pede para um patologista que acompanha os transplantadores, né, que pela forma eles fazem uma
conseguem
microscopia eletrônica do glomérulo, para você ter uma ideia de quais regiões do glomérulo estão mais afetadas, e daí sacar o mecanismo. Então, isso é bastante promissor.
Obrigada, professor.
Até porque... conversa.
Não, adorei. É muito legal, porque a gente acabou vendo mecanismos que são independentes e correlatos, né? O que acontece no túbulo não parece ser o mesmo que acontece no glomérulo. Porque o que se acreditava é que, antigamente, se você pegar, a literatura é muito antiga, né, em doença renal associada à malária. Então, o que se acreditava é que, primeiro, você tinha um dano glomerular, e por isso as células do sistema imune, na verdade, elas invadiam para a região tubular, mas porque o glomérulo estava danificado. Não se acreditava na verdade, a chegada dessas células pelo interstício. Isso é realmente mais recente na literatura. E tem já relatos que essas células estão realmente em contato com as células tubulares, principalmente células T, mas especificamente o mecanismo, isso está muito mal descrito na literatura e é um nicho realmente para ser investigado. Agradeço muito suas sugestões. Esse trabalho da doença renal eu faço em colaboração com o professor Celso Caruso, né? E a gente tem uma colaboração muito próxima. E ele ajuda bastante, né, toda essa parte de lesão, de doença renal. Então, esse é um dos modelos, né, de doença renal do laboratório, mas, na verdade, a gente tem vários modelos diferentes, inclusive modelos estudando uma doença ainda, um estágio antes, né, de virar acute kidney injury, no caso a gente chama de sub-AKI, porque o laboratório, inclusive o grupo dele, busca muitos marcadores, né, que são preditores do dano renal, da lesão renal, na tentativa de impedir que isso evolua para os quadros mais graves, né, da doença renal, que chega a ser importante aí a questão do transplante, aí são pacientes que vão requerer realmente uma terapia mais, né,
sim, claro,
severa, enfim. Então, eu agradeço muito mesmo as suas dicas e os seus comentários. Muito obrigada.
Parabéns, e nós vamos nos encontrar via Júlio, provavelmente.
Com certeza. Ah, que bom. Isso aí.
Prazer, muito obrigado.
Foi um prazer.
E desculpa falar em português, mas quando eu falo em português, eu falo com o meu coração. Entende?
Eu sei, não, é porque tem coisas que a gente só consegue expressar na nossa língua mesmo.
Muito obrigado.
Obrigada.
Obrigada, professor. Não, a gente faz em inglês porque tem alguns alunos da USP que são estrangeiros e terminam se comunicando entre laboratório e inglês, mas agora, né, nesse ponto da discussão, acho que já vale uma coisa mais acalentadora mesmo.
Mas
não tem problema nenhum.
De expressar a minha emoção e o meu entusiasmo pelo tema, tá certo? Ainda mais envolvendo o meu amigo Júlio.
Que bom.
Sim, com certeza.
Essa oportunidade agora de conhecer de perto, e eu acho que vamos ter colaborações seguramente, todos os peptídeos, todas as proteases sempre passaram aqui por São Paulo, e vamos trabalhar juntos seguramente.
Tá certo.
Muito obrigado.
Obrigada. Um abraço, professor. Obrigada.
Obrigada, professor. Obrigada de novo, Ana Cássia, pelo seu tempo, pela discussão extremamente frutífera e interessante demais, né, Cris?
Gostei muito.
Obrigadíssima, Ana Cássia. Foi sensacional. Temos muito que conversar agora, mais do que antes.
Temos. É isso aí.